Okay, so last we were talking about, we had talked a little bit about the different options. So we know that the options go together with the retinal to actually form rhodopsin. Okay, so these opsins, these are these proteins and everything. So there are three different opsins. Each opsin is responsible for recognizing a certain color in white light. But when we talk about the photons, it's actually recognizing a wavelength of a certain photon of white light. So if we take a look at white light can basically be broken down into the spectrum. You guys see it below here. You see it below here at the very bottom. So the violet, the blue, the cyan, the green, the yellow, and the red spectra. So all of those colors basically make up what white light is. And so white light could be diffracted into each of those individual colors, and then of light. And then those individual colors of light are at different wavelengths, and that represents that particular color. So the opsins are able to sit there and, I guess, capture certain wavelengths. And so we have the blue opsin, and it's responsible for, it's out in the blue cones. It recognizes the wavelength that is at 420 nanometers. The green opsin recognizes the wavelength at 534 nanometers, and the red opsin recognizes the wavelength at 564 nanometers. And so these guys are going to be in our cones, right? And so you have cones that are dedicated to recognizing that particular wavelength of light. Okay.
Mm-hmm.
And so the rods, on the other hand, so rods are going to be sensitive to light, so that are going to be so sensitive that basically any wavelength of light is going to be, or any photon is going to be able to sort of activate it. Where the ones that have the specific colors, they're going to only be activated under their specific colors. Okay, so does that make sense?
So like different shadings of light triggers different cones?
So different wavelengths of light triggers different cones.
Oh, okay.
Yeah, where the rod is just going to be triggered all the time in the case of any wavelength of light, where only certain cones are going to be triggered by certain lights. So you can kind of imagine, so a person who's maybe colorblind is a result of a defect in one of those cones.
Mm-hmm.
Okay, so this here is the sensory pathways of the spinal cord, and so I'm going to type this up because it's really hard to see. One day I'll get my stuff together that I'll have all my slides processed so you can actually visualize it. In the book you can kind of see it, but it's still kind of hard because it's tiny.
Mm-hmm.
The images are not the best, unfortunately. So this is the medulla, this is the middle brain. This guy's it's a second order nerve, or sorry, second order neuron. third order neuron. Come on. I don't know why it's not changing for me. Okay, there you go. Okay, so down here we have our spinal cord, and if we look, this here represents the dorsal column system. This here represents the spinal thalamic tract. So in the case of the dorsal system, so if we get a touch, so a touch is kind of like a very fine touch. It's going to basically trigger a basically impulse going from a dorsal ganglia root. There's a synapse. Synapse is going to be attach to that first order neuron. It's going to go through that spinal cord, and then up through the facialis conoides, and then to the facialis gracilis, up to the nucleus cilius, and then it's going to actually connect to the second order neuron, and then the second order neuron is then going to go to the thalamus, and then at the thalamus it's going to connect to the third order neuron. So if we kind of think about it, so these neurons are pretty long in length in terms of the fact that you're going down your dorsal column system and you're entering that painter's through that dorsal root ganglia. And then that's going to take us, which is going to, take us to that first order neuron. That neuron then connects to that middle neuron, which is that second order neuron. And then that one connects to that third order neuron. Okay, so in the case of the the spinal thalamic tract, so that guy is basically going to be going, and this is going to be our pain and temperature sensations. So that one is also going to go through that first order neuron. Then that's going to connect to that second order neuron, and then to the third order neuron. And so the only distinction is the location of those neurons in the different regions, right? So, and then that basically takes us, that third order neuron takes us basically to the brain, where we'll be at which is located at the postcentral gyrus. Come on. Okay, so when we're thinking about the mechanism of sound, so in this case, if sound is coming from, let's say, on the left-hand side, like in the lower. So what's going to happen is that the auditory system is going to basically try to come. So that ear, our ear on our left-hand side, the sound is going to basically arrive there first, and it's going to be louder. And then the ear on our right-hand side, sound is going to arrive there later, and it's going to be softer. And so the connection between the neurons on either sides are going to be used for comparison. So they're going to use to compare the sound from one side to the other side to kind of get a difference in that intensity. Mm-hmm. Also in terms of time. And so this is called the inter-aural time and intensity differences. So the neurons are responsible for recognizing that. So if sound is coming in evenly, right, from both sides, it has the same intensity, it also recognizes that as well too. So those neurons are responsible for recognizing that as well too. So we get a comparison to see whether or not things are, even on both sides, or whether it's to the left or the right, And so if it is, we hear it from here, so our response is hear a loud sound that comes from the right-hand side, our instinct is to look at it because it recognizes that the intensity over here is greater than what we hear on the other side.
Okay.
Okay. Okay, so in terms of our eyes, so as we turn and rotate our head, our eyes are going to kind of compensate for that rotation. Okay, so if we're turning towards the left, then our eyes kind of turn towards the right to kind of compensate for that movement. And so it kind of goes in opposite directions. So the eye muscle is naturally going to go in the opposite directions to kind of compensate for that turn to that particular direction. Does that kind of also make sense?
Yeah, because like they're fixed on a location while we're turning our head.
Yeah.
Okay.
Okay, so in terms of our visual field, so our eyes are kind of divided. So one part of our eyes kind of recognizes, let's say, the right-hand side, and then the other part of their eyes recognizes the left-hand side. And so if you take a look at our right eye, so this basically is the binocular field. So if we take a look here, so then in our eye kind of inverts it, goes through and invert those sides. So the left side of the vision is going to be going towards the right side in the inner part of the eye. So then that is going to be transferred to, in this case, the right side of the head. The left side is going to be transferred to it's going to be on the opposite side because we see things sort of opposite of what they are, right? I guess. Let's see, I should say, not that we see things. Our brain is transmitted opposite to what side they are.
So the images we see on our right eye go to our left brain?
The images that we see on our right side of our right eye goes to the left side of the brain. The images that we see on the right side of our eye goes to, because in a sense, you have connectors on both sides. So like if you look, close one eye. If you close one eye, you can see the full spectrum. It's not like you don't see the full thing. So you see both the right side and you see the left side. There's like a limitation to that field of view, right? There's only so far it can go. And then the same thing happens with your left eye. You can see on the right-hand side and the left-hand side, but the field of view is going to be a little bit different. And so, and then you have the one where you see you have overlap between the two when you have both your eyes open.
Mm-hmm.
Okay. So, but again, it's kind of divvied up. So part of that signal to that eye is being sent. So the one that's on this side is going to go on my left-hand side. It's going to go to the back side of my right brain. The one that's on this side, one that's on this side is going to go to my one that's on my, so the vision that I see here is going to go to this side of my brain. The vision that I see here, and then right here in this case, it's going to go to this side of the brain. The vision that I see over here is going to go to this side of the brain, and the vision that I see kind of in that inner part is going to go to my left-hand side. So the left is going to go to the right, and then the right is going to go to the left. Does it make sense?
Yes.
I was struggling with this earlier today. Okay.
I guess it makes sense so you could get a full complete picture. Even if you only have one eye, you don't get like fully blindsided.
Yeah, that's right. And then, and again, like I said, if you only have one eye, you still have that sense. You can see the full thing, but it's not as clear as when you see it with both eyes.
Mm-hmm.
Okay. Let's see. Okay, so here we're talking about the cortical processing. So this is just basically showing us, so this word here, this word means little man. And so this basically shows you in terms of that cortical region where all of the different processing is going to be taking place. So anything that deals with my genitalia is going to fall in this area. My toes, my feet, my hips, my legs, my hips, it's going to fall within this region. My trunk, the neck, the shoulders, the arms, it's all going to fall here. Okay, so that's where the processing for those events or those pain or touch sensations are going to come from. That's where all that processing is going to, well, being delivered to, not come from, but being delivered to. Okay, and so the take-home message from this cartoon is just, different regions of the cortex are responsible for processing different parts of the body. So we have those signals being delivered up to those parts of the body. This here is a topical mapping of the retina. And so if we sit there, we look at an image. So in this case, they have this nice little image of a house. The upper left field is going to be the roof of the house. The lower left field is going to be the house itself, right? So our eyes kind of break this part down. So it actually will reconstruct the image, but if you take a look at the coloring, so the purple represents what is being observed by the purple region. So all of that is going to be processed and put to the right side of our head, where the stuff that's in the green region is going to be processed and put on the left side of our head. And then the same thing over here with this eye. So then in this context, we're getting parts, right? So we got this part here and we got this part here that are being processed, but you're not getting the full thing. So what happens is that when we go to the brain will reconstruct that topical image. And so it'll actually break things up so that we can actually reconstruct that image. So we'll have that lower field and the upper field. And so our brain, so the upper left visual cortex it's gonna process basically the upper right component. And so you end up with this image being here. And then the upper right, or the superior right visual cortex, is going to process this part here. And the lower components are going to be processed simultaneously as well too. So it's going to reconstruct that full image after it's broken them up. And so you can kind of imagine if you may have some type of damage in one of your cortex, it could actually affect what you perceive and what you actually see. So you may be missing an entire region as a result of the fact that your cortex has been damaged within that area, right? So let's say that the superior right cortex is damaged. So then you're going to be missing that upper region. You're going to be missing the roof. So you won't be able to see when the roof is on fire.
Mm-hmm.
Okay, so do me a favor. Put your finger in front of you and look at it with one eye. Your right eye. Funny. Cover your other eye.
Okay.
Okay. Now look at it with your left eye. Covering your right eye. Okay. The distance, the depth perception, did it change when you looked at it between the two different eyes?
Left eye is a bit farther away and it moves more to the right.
Okay. Okay. So what happens is that with both of our eyes open, we actually kind of get a more accurate sense of the depth. One eye is going to go too far and one eye basically goes a little short. So that depth perception of one eye is going to be a little bit different than what we see with the other eye. One's going to be a little closer, one's going to be further away. And what happens is by us opening both eyes, we get a more accurate depth perception than when we see with one eye. So if we look at something far away, do the same thing. Cover one eye, cover your right eye, and then look with your left. Then cover your left eye and look with your right. Does depth perception change?
Yep, definitely.
Okay, so now, have you ever gone to a movie, a 3D movie with the glasses?
When I was really young, yeah.
Well, you don't do that anymore. Well, I guess not anymore because of COVID. You know, nobody does movies. So, but you don't go to any 3D movies?
They stopped showing them around a couple of years ago. I think the last one was Avatar with the blue aliens.
I saw one of the Marvel movies in 3D.
Was it Spider-Man?
No, it was Captain Marvel.
That was 3D.
In some theaters, it was 3D. I saw that one. Okay, so 3D glasses kind of take advantage of that. So because it's playing with the light, and then also the our perspectives, if you kind of notice, you have one side that'll be blue and one side that'll be green for your 3D glasses. It adds that third dimensional to it. So it gives it that kind of spacing, that little more depth to it. So it's no longer a two-dimensional image that we're seeing, we're actually seeing that three-dimensional image, you know.
Like I know when you take off the glasses, the images are sort of spaced
Oh,
out.
they're blurred. Yep. Yep.
It's kind of like when you're closing one eye, your finger looks more to the right, and then you open the other eye, is more to the left.
Yep.
But then when you open them, we put them together, it's all one image. Is that how 3D glasses work? By making the image
Exactly. Yeah.
one?
Yep. Yeah, because it amplifies, so that one lens will amplify it more in one case and amplifies the other in the other case. And so it puts that image in more of a 3D plane rather than just that 2D perspective.
Mm.
So in a lot of ways, you're tricking your brain. So something that would be normally two-dimensional, that flat screen, is now looking as if it's three-dimensional. Oh. Okay, so if we sit there and we think about the streams of stimuli, so when we're receiving those stimuli, um, the visual stream that is going to come out. So depending on what it is that we're picking up. So like for instance, the dorsal visual stream is going to help us determine where it is. So that kind of lets us know, okay, where is it located at? Is it on the right hand side or is it on the left hand side? Okay, and so the dorsal visual stream, so which is going to go from the occipital lobe to the parietal lobe. So in this direction, that's going to sit there and determine location. And then in terms of when we're trying to figure out what it is, right? So that's going to be going from the occipital lobe to the temporal lobe. And so that's going to be your ventral. So the upper part of your brain is going to sit there and determine, okay, location. Where is it located? It's going to give us regions where it's located. And then to kind of get the clarity as to what it is, okay, it's a deer, right? So then the ventral part of our brain, that lower half, is going to sit there and let us know that component. And so again, if we have some type of damage, it could actually affect that determination of, okay, what is it? Compared to if another part could sit there and have issues with location. Is it on the right or on the left? I can't tell. Okay. Mm-hmm. So there's this guy named Phineas Gage. He had an accident where a rod, and this is him standing next to the rod, that went literally through his cheekbone up to the top of the skull. He actually lived through the process and everything like that. But this had been shown to actually have an effect on his emotions, right? And so they said that, like, before he had the accident, he was very amicable. He was a hard worker. And then all of his friends afterwards said that, after he had that accident, he wasn't quite the same. He was very irritable. He was temperamental, and he was lazy. And so he went like from night and day to the two components. And so a lot of his friends basically said, oh, it was kind of like, how like when you get really drunk or you're on drugs or something like that, if you have too much, you become a little belligerent. So like you've heard about the guys that always get into black bar fights when they get drunk and stuff. They get there now, it all worked up. Or if they're like on meth, Okay, so the regions of the brain that were actually damaged in this part were they were the prefrontal cortex of his frontal lobe, and so it's also shown that that's the part that's stimulated after so much alcohol or so much drugs, and particularly people who have anger issues, right? So so this here is just the cortical spinal tract. So if we take a look here, and this is kind of cool. If you think it's pretty, I think it's pretty cool because you kind of get an idea of the neurons that are involved in this. And so these neurons are pretty long because these neurons are going to be going from, let's say, the skeletal muscle in the case of the lower neuron. So we'll have this lower neuron here that is going to our skeletal muscle. That is going to be connected to the upper motor neuron. These upper motor neurons are going to they're going to extend all the way from the point of connection in the spine. And they're going to be going to their, I should say, their cell body is going to be located in the frontal lobe. Okay, so literally they're going to be expanding. That axon is going to expand all the way from our brain to, let's say, here's like the cortex of our, I mean, here's the our spinal cord. So all the way up to the spinal cord where they're going to be going to that skeletal muscle, to the periphery. So like say on our arm or anything like that. So we have a neuron that basically travels from there all the way up into the cortex of our brain. So that kind of gives us an idea of the length of these neurons, these motor neurons, these upper motor neurons. Okay. So questions, concerns, Cash?
There's literally a neuron from our brain all the way to like our toes to control them.
It's not all the way to our toes. There's actually, so it'll still be in the skeletal region.
Mm-hmm.
I mean, not the skeletal region, in the spinal cord. So at the base of the spinal cord, there'll be one that'll be going down towards our toes. Yeah.
Oh.
So in our spinal cord, so like as we go down our spinal cord, there are going to be neurons that are going to the periphery. So, but spinal cord is going to be connecting. There's going to be a axon traveling along the length of our spinal cord, where things are going to be connected. Does that make sense?
Yep.
Okay. So, but it's amazing that in that region now, it's like your cell body is all located on one. So, you know, the nucleus, the organelles are all located there, but the actual cord itself, you know, I mean, the axon is being shot there. And in some cases, we have connections that'll go to the other parts of the periphery, but we have this particular axon, that upper motor neuron, that's kind of connecting to that lower motor neuron, right? And so it's composed of just two neurons. If you think about it, you have the upper one and you have your lower one, right? And so you think about it, if it gets screwed up, let's say in the case of, you know, a paraplegic, that kind of explains the issues that we have in the case of a paraplegic. So depending on where they're hit or they're injured, because that neuron is so long, that motor neuron is so long, then you basically damage that individual. It's not like there are components that can be replaced and repaired in between.
Hmm.
Okay. Okay, so that is chapter 14. We're going to go over a little bit of chapter 15, and then we'll head out. Made it to this. Okay, so we're going to be looking at autonomous nervous system now. So the autonomous nervous system is kind of responsible for our fight or flight response. And the autonomous nervous system can be basically broken up into two divisions. We have the sympathetic division, and we have the parasympathetic. So the sympathetic division that's going to be the actual fight or flight. The parasympathetic division is going to be kind of like our rest and digest. So that's like our chill. Eat a nice meal and chill mode, right?
Mm-hmm.
Okay, so in the case of our sympathetic division, so in that case, we're getting that stimuli. That stimuli is telling us, okay, you have this predator, it's coming after you. You're going to get all worked up. And as a response, you're typically going to do a couple of things, right? One of the things that typically happens under these circumstances is that your pupil will dilate. You'll start getting sweaty. You'll start getting kind of worked up, right? You're not hungry. You're not thinking, okay, yeah, I can eat right now. You're thinking, I need to run, or I need to, or I need to fight. Your adrenaline starts flowing, Is that right? Or am I making it up? Is that just what happens to me?
Yeah, you're right. Like you start breathing heavier.
Yeah, you start breathing heavier. Yeah.
I think maybe if it chooses flight, your muscles start asking for more oxygen.
That's right. And so in that context, right? So you're breathing that oxygen and everything like that. But let's say your stomach, you no longer feel hungry, you're not getting the oxygen down there for food digestion because you don't need it. You're thinking about using the muscles. And so you're sending the oxygen towards your muscles so that they can respond, and then they can start processing that glycogen that they have stored in there and start using for energy that you're going to be needing to run away from that predator, right? Okay, so back in the day, we had to worry about, being hunted by other animals, dinosaurs and let's say lions and tigers. And then today, we don't have that problem because the way our society is, but we still trigger those same responses, so instead we'll trigger it. And when we're talking to a girl that we really like for the first time and you're going to ask her out, right? What happens when you do that? Or talking to a boy that you really like that you want to ask out,
The heart starts beating real fast.
Yeah, your heart starts beating real fast. You start sweating, right? You're not thinking about food. You don't want to eat. You're just like, all you're thinking about all the nerves that are building up on the inside,
Mm-hmm.
right? So that anxiety, right? So, and that's the same thing like when we get talks, where we talk about that rest and digest, you're just kind of, you've already got the girl and everything like that. You guys are having dinner. This is like you've seen her all the time. And so you guys are just kind of kicking back the meal and just, and all you want to do is sleep,
Mm-hmm. Just going with the flow.
That's right. Okay. So all of this is controlled by your autonomous nervous system. The fight or flight component, the symbiotic nervous system. So this is just basically those ties that are tying it together. These are the neurons from the lateral horn of the spinal cord. And so you can kind of see the lateral horn of the spinal cord right there. And, of course, it's going to be on both sides of the vertebrae, depending on the right-hand side or the left-hand side, who it's going to be tied to. And then these guys are going to be connected to other ganglia. Right? And so so you're going to have basically going to be projecting to different chains of ganglia on either side of your vertebrae, so depending on the side that you're on, and then or to the anterior or the vertebrae columns of the abdomen cavity. Okay, so then the exons from these ganglionic neurons are then going to be projecting to the effectors. So these guys here are then going to go and project to the effectors. So we have that interconnection in the spinal cord, and then those guys are going to be connecting to our targets. So those are going to be like our organs. Okay.
Mm-hmm.
And so this basically is the kind of like the mapping of the neural system where they belong to. So in the T1, we'll be going to the heart and the larynx and the esophagus, right? And then you can see that that's T1, T2, T3, T4. So all of those are all connected in that particular area. And so we can kind of see how the organization from the spinal cord up to the point where we're connecting to the effector organs as well. Okay. So in the case of, so the axons from the center sympathetic neuron and the spinal cord, they can be projected in a couple of different ways, right? One of the ways is fibers can be projected out to the ganglia at the same level, and then to the synapses of the ganglia neurons. So if you take a look here, I know you can't see it very well, but like in the case of T1, you have, here's our spinal cord, and you can actually see the synapses forming between that ganglia and then the ganglionic neuron that is going towards, in this case, another organelle. Okay, and then this is a closer and more anatomical vision of it. So spinal cord, so we're going from the spinal cord to the spinal nerve, and it's going to be connecting in and this is the white ramus communicans, so this area. And then this is where we're going to get it connected to the synapses. So this is where the synapse is going to occur to connect from the ganglion to the ganglionic neuron of the effector or our target or organ. Okay, B, we could have, so A was like a fiber can project. The next part, we can get branches, right? A branch can project to more superior or inferior ganglion in the chain. So in this case, we have this guy forming synapses in different areas, forming like a little branch. And so then these guys could sit there and connect to a couple of different organelles, all coming from that same axon. So that axon is just basically breaking up into little branches. And so it has little dendrites on each end, forming those synapses. Okay, and so here you can see another picture. Going from the spinal cord, here's your spinal nerve. So that spinal nerve is now connected to one of the target effectors. So that would be like this guy, it's connecting to one or the other. But you could have multiple areas where this guy is branching off. So we could have another connector that would branch off at this point, or another one that could branch off at that point, all going to that same, that all being the same axon. Okay, or we could have branches that are projecting through the white ramus communicon, but not terminating on a ganglionic neuron chain. Instead, it projects through the splenic nerve to a co-lateral ganglion, or, which is not shown, the adrenaline with the medulla. So in this case, we have it coming from the spine. It continues to go beyond that, and then it branches off. Okay, so here, continuously through the spine, it goes through the sympalachnic nerve, and then it goes to another, another, in this case, the prevariate ganglion, and forms the synapses there. And then that takes us to the thing. So this guy extends even further out, and you're also getting several branching components. Okay. So with the parasympathetic, so these guys are going to be at the brain stem. So these are neurons that are going to be found in the brain stem or in the lateral horn of the sacral spinal cord. So then, in other words, the upper part of your neck in this area here. So the spinal cord there. So in this case, these axons, they're going to be basically, they're going to project short distances to target effectors. And then occasionally we're going to have a few that are going to be located, here at the lower part of the horn. And so that's going to deal with those areas that are further away. So large intestines, the rectum, things like that. Okay. The upper part of your spinal cord, you'll have a few long nerves, but for the most part, they're going to be targeted shorter distances. Okay. So like, for instance, the cranial nerve 3, so that guy is going to be connected to the ciliary ganglia, and then that's going to be connected to the eye. And so where cranial nerve 10, that guy is going to be connected to like our internal intestines. So all the stuff that's down here. Okay. And so again, those nerves are going to be responsible for like more of that resting, digesting when we're not kind of worked up. Where these guys here in the red, they're going to be responsible for that flight and fight response. And so you can kind of see you have, when you do have these guys, they're typically they're long, you know, I mean, not long. They're typically, how do I say this? They're not long, but their projections are not that short and concise between one to the next to the next. So in this case, it's like you're given time for response, where in the other case, you have your connections are kind of located in a shorter intervals with each other, and also you're getting quicker responses as a result of that location. So like here, you can see that it's more distributed throughout, and then the targets are all leading towards that spinal cord, and so that you're getting that quicker response to, or quicker signaling to give quicker responses. Does that make sense?
So basically, the sympathetic is there in the moment.
Yes, because in the case of flight, if you have to take a time to sit there and do it, then you may not survive, right? So that guy, its organization is more short, quick, you know?
That way you can fire away the signals faster.
Exactly.
Mm.
Okay. What time do we end? 2:30, right?
Yeah.
Okay. So you're doing it again. We're still early. So we stopped here in the other classes, so I'm going to stop here too.
Gives me more time to finish the lab.
What was that?
That gives me more time to finish the lab then.
Yeah. Okay. And as I was saying, we'll be finishing up. We have 4 more weeks left.
How many more chapters?
Well, a lot.
Really?
Yeah, we do. We have a lot. But the thing is about a lot of the chapters, some of it, and I'm in the process of kind of cleaning them up a little bit. Some of them have mixtures of anatomy and like heavy anatomy and physiology. And so I'm kind of cleaning them up. I kind of did that. I showed you when I showed you the chapters that we have.
Yeah.
So we have like next week we'll be doing 15, finishing 15, 16, and 17. Then and part of 18. Well, part of 16, part of 17 is well, 17 is the endocrine system. So we'll be talking a lot more about the hormones, and it's going to be a big chapter with physiology, because it kind of triggers and control aspects of it. But there's a lot of it is also anatomy. So we'll be kind of cleaning up that anatomical parts. You know enough to kind of get us through. So there'll be a couple of slides, so that'll be a lot less.
Okay.
Yeah. And then in the case of like chapters, the week after that, we're going to be doing chapters 19, 20, 21, and like big parts of 20 is going to be missing because a lot of it's anatomy, and big parts of 19 is going to be missing a lot of part of anatomy. We may be able to squeeze 22 in there. So we're going to be doing like 2 chapters a week, or 2 chapters each lecture.
Oof. All right.
Yeah. Well, the meat and potatoes is like chapters 24 for this class. And that is, but we need all of the other ones because it plays a little bit of part of everything, but chapter 24 is metabolism, and that's physiology. You know, that is a large part of physiology. So we'll get there.
Easy.
And it's funny because I think about it, when I took physio and anatomy, I took the metabolism part first, and then all of the muscle and stuff like that, how everything pertains to the particular muscle afterwards, which is kind of weird that the book has it at the end rather than the beginning.
What made that change?
Yeah. So I'll have to, what was that?
Probably just easier to print, save money.
Yeah. Or it was something that, you know, it was an afterthought, you know, like, oh, we're going to do all of it in line, but there's like large parts, like in terms of metabolism, we talk about cellular respiration, we talk about absorption, and then all of the things that are responsible for things, right? And the components, and so yeah, the meat and potatoes, chapters 24 and 25. That's a large portion of anatomy. I mean, not anatomy, physiology. So 24 is, like I said, metabolism and nutrition, and then 25 is the urinary system. Okay. Any questions of how studying for the test going?
It's going all right. How the cheat sheet, is it just front or can it be front and back?
In this case, it can be front and back.
Okay. Wait, in this case, does that mean the final is only be front?
No, the final is going to be front and back.
I was worried there for a sec.
Yep.
Are you gonna be available for Friday at 2?
Yes. Yep.
Do I just click on the normal lab session?
Shoot me a text, and then to remind me, and then click on the actually let's do my office hours.
2:30 then?
I said go to my office hours.
It's only Thursday.
Well, yeah, I know, but when you click on, you can click on it on Friday as well. And then when you click on it on Friday, if you text me, I'll be there, and you'll be able to come in.
Yeah, okay, I'll do that. I'll text you or
Okay.
send you an email.
Okay.
That's all the questions I have. So thank you for the lecture.
No problem, and I'll see you next week.
All right. Have a good day.
You too. Bye-bye.